Today we're going to discuss deoxyribonucleic acid, i.e. DNA structure. We will also talk about the basic 3D structure of DNA, its components, types, and the double helix structure of DNA. We all know that we get our hereditary material, i.e. DNA, from our parents. Have you ever wondered what it looks like? The structure of the DNA is like a ladder. Let's find out how. In 1953, Watson and Crick proposed that deoxyribonucleic acid, DNA, has a right-handed double helical structure which has two strands of polynucleotide coiled around a common access point and held together by the hydrogen bonds between the bases of the complementary strands, aka bDNA. The hydrogen bonds are formed between purine and pyrimidine. Remember, as we can see in the diagram, pyrimidine is a six-membered heterocyclic ring with alternating single and double bonds, whereas purine is a compound fused with pyrimidine and a 5-carbon compound ring. Now let's dive deeper. DNA is a polymer of polynucleotides, where the phosphate diester bonds join the deoxyribose sugars at the 3 and 5 hydroxyl groups of the adjacent nucleotides. The backbone of DNA comprises 6 single bonds that help in flexible rotation and give possible structural and conformational changes to the DNA strand. Each turn of the double helix of the DNA consists of 10 nucleotides separated from each other by plus 36 degrees rotation and 3.4 A translation along the axis. Remember, these two strands run anti-parallel to each other, i.e., one in a 5 to 3 direction and the other one in a 3 to 5 direction. We have discussed bDNA. Now let's discuss the A and Z forms of the DNA structure, which are slightly different from the B form. As seen in the table, A DNA is the thicker right-handed helical structure, where the distance between the base pair is less than the bDNA. Thus, A DNA includes more bases in a single helical turn. Next, we have the Z DNA, in which the rotation of the helical strands is left-handed and includes 12 bases in each turn. It is formed from the alternate purine and pyrimidine stretch and gives a zigzag structure. Moving further, let's understand Chargaff's rule, which explains the base pairing. In this, the amount of adenine is equal to the thymine, and the amount of guanine is equal to cytosine. Thus, a 1-to-1 stoichiometric ratio of purine and pyrimidine bases is stored in every organism. The two DNA strands are held together by the AT and CG base pairing, which gives the double helical structure similar to a ladder. It is often incorrectly assumed that in Chargaff's rule, the ratio of purine and pyrimidine is the opposite. That is, adenine is paired with guanine and thymine is paired with cytosine. This mistake leads to the incorrect Chargaff's ratio because DNA strands are held together by the AT and CG base pairing. To sum up, we now understand what our DNA looks like. We have also learned the basic 3D structure of DNA, its components, types, and the double helix structure of DNA that is similar to a ladder.